question and answer session at the end of this presentation. To do so, in the bottom right-hand corner of your screen that you see, you're going to see a Q&A section. Simply type in your question there and hit send, and that question will be delivered to us, and again, we'll be answering them at the end of the presentations. If for some reason you do not see that Q&A box in the bottom right-hand corner of your screen, at the top right-hand corner of your screen, you're going to see several icons. Simply hit the Q&A, and that will bring up that box for you. For everyone that did register for this event, we will be receiving separately copies of the presentations, copies of the Q&A, as well as some supplementary information from both Agilent and Milestone. Now clearly this USP, the new methods coming out for USP, is a pretty hot topic. It's been around for a long time, and it's had some delays and some changes, but again, clearly it's something a lot of people are interested in, given that we have several hundred attendees to this event. And what we're really hoping to do for you all through these presentations is to, one, provide you a clear picture of the important implementation dates as they stand today and requirements of the new methods. We want to give you a better understanding of the analytical component of the methods, as well as the sample preparation technology that's available. And also, because one interesting thing about this is that metals analysis is new for a lot of labs in your industry. And so there's a lot of information out there, a lot of different things. So really what we try to do is provide some key considerations, to make as you think about beginning to implement these methods. So with that being said, I am going to pass the ball over to Steve, and we'll go from there. I'll be talking to you soon.
From the outline, we'll first review the existing USP Heavy Metals Chapter 231. Next, we'll talk about USP Chapter 232, which lists elemental impurities limits. The ICH Q3D draft guidelines will be reviewed, including harmonization status with USP. And then we'll examine USP 233, which specifies procedures. This will include a look at the two compendial techniques, Icp-Oes and Icp-Ms. Included here as well will be a review of the J value, where we'll actually go through some specific calculation examples. We'll also talk a bit about electronic records and compliance solutions, and we'll end up with an update on the latest implementation timetable. But first, let's discuss quickly why it's important to be able to determine elemental impurities in drug products. As many are aware, the presence of elemental impurities can potentially have adverse health effects. This depends on which elements are present and, of course, at what levels. So that's why limits have been established for permissible amounts of elemental impurities in drug products. Elemental impurities may include things that are added during the manufacturing process, such as metal catalysts. They could come from raw materials, could be impurities from production equipment, or it could be present in excipients or other inactive components. So USP 231 is an existing test for what were classified as heavy metals. The test indicates the metal impurities using a colored sulfide precipitate. And as you can see, this test has been in use since 1905. It's not element specific. It's a visual or subjective comparison test. And the standard solution the color can change over time, which results in poor reproducibility. So there's issues with poor recovery as well when the ash sample prep procedure is used. And in some cases, depending on the procedure, relatively large sample sizes are required, anywhere from 1 gram up to 5 grams, which isn't often convenient. So bottom line, it's a subjective test. It was developed before the advent of modern analytical instruments, yet it's still in use today. So it's long been known 231 is a relatively ineffective test. And here we can see a graphic from an often referenced paper by Nancy Lewin and others that visually illustrates the limitations. So if we take a look at this graph, we can see along the x-axis, we can see the different elements of interest, and on the y-axis, we can see the percent recoveries. So if I can draw your attention to the striped bars on the bar graph, we can see that for most of the elements, and those represent the 231 test results, see that most of these elements are less than 50%. Lead is less than 50%, arsenic less than 50%. A couple of elements are down by less than 10% recovery. In fact, if we look over here at mercury, we have approximately a 0% recovery. So 231 is not a very effective test, and in this case, it's due to the ashing step, which gives low recoveries for certain elements. But if we apply modern technology, in this case using Icp-Ms, we can see with the black bar that if we use Icp-Ms technology in appropriate sample prep, that we can actually get very good recoveries, and that's what you see with the black bar. So that represents the recoveries for Icp-Ms, all pretty close to 100%. So the study was published in 2004, and a short time later, USP appointed an advisory panel with both experts in methodology and toxicology. So just another statement here, this one from USP showing the commitment to advancing 231. So USP meeting dated from April 2009 says that we are committed to advancing the current standards so that widely agreed upon safe limits for key metal impurities are properly measured, thereby protecting the public health. So this is really a clear indication of acceptance that current method 231 does not adequately protect the public health. So USP 231, as most people know, is being replaced by two separate chapters, 232, which addresses impurity limits, and 233, which addresses procedures for carrying out the testing. We can also see the terminology was changed from heavy metals to elemental impurities. So here we can see the official, but not yet implemented 232. So this document lists the elements, the limits, in terms of PDEs, or permissible daily exposures, and the limits are based on the route of administration. So the route of administration is just a dosage form, so for example, oral, parenteral, or inhalation. And it's also important to note that 232 is a risk-based method or uses a risk-based approach. So while the limits are given, the frequency of the testing and actually the specific elements that are required to be tested are determined by the user's risk-based protocols. So that's an important distinction. Another important distinction for 232, and this is often misunderstood, is that 232 applies to drug products. So when we're talking about drug products, this is not applicable to drug substances or excipients. So one reason there's a lot of confusion around this is that there's going to be a Table 1, which lists the impurity levels for drug products. But in Table 2, it looks like there's limits listed for drug substances and excipients. So that's the main reason why there's confusion around what's actually covered by 232. But the idea of having Table 2 in there is that elemental impurities in drug substances and excipients, they should be known and they should be controlled so that drug product manufacturers can use the information. But again, they're not held to the same limits as drug products. Interesting, in some cases, the elemental impurity limits may actually be exceeded in drug substances or excipients, and in some cases that's okay. So for example, if you're a drug product manufacturer and you're only using a very small amount of a specific ingredient in the final dosage product, maybe it's going to be okay if in the excipient the actual elemental impurity level is exceeded. So the chapter also outlines options for demonstrating compliance. There's the drug product analysis option and also the summation option as well. It goes. Okay. It should be noted that revisions to Official Method 232 were proposed by USP in December 2013. That's what we can see here, the revised document. We'll talk more about why these revisions were proposed, but first, let's talk about what the revisions are. So to do this, we'll take a closer look at Table 1 and Table 2. So this is from proposed revision of 232. And for Table 1, what we can see across the top is that we can see the route of administration. For example, oral, parenteral, or inhalation. And then along the side, we can see the different elements: cadmium, lead, arsenic, mercury, etc. down the side. So USP did actually a really nice job in the revision, because anything that's been changed, or I should say proposed to be changed, is highlighted in yellow. Not only is it highlighted in yellow, but the previous value is still shown, it's just crossed out, and you can see the proposed value next to it. So for example, cadmium, if we look at this, we can see that cadmium was 25. In the new proposed revision, that value is changing to 5 micrograms per day. So you can see there's quite a few changes that are proposed to the limits. Over here in Table 2, we can see the default concentration limits, and these are for the drug substances and excipients. So in this case, there was a 10-gram sample size used. So you can see that basically what that does, it moves over the decimal place one time. So we can see that everything is about 10 times lower for the example of a 10-gram daily dose. Okay, so now that we know what some of the changes are, it's also important to know the reason for the revisions. So we can see that on July 2013, ICH releases the Q3D Step 2 document. So I should mention that ICH is the International Conference on Harmonization, and this ICH attempts to harmonize global standards. And this can be important as most drug products are marketed around the world. So US as well as European and Japanese representatives are all involved with the ICH. So based on the ICH document, USP decided to propose the revision to 232. So as we saw, some of the ICH limits were adopted, but some were not. So what's called now is a partial alignment of 232 with the ICH document. So when USP came out with their 232 proposed revision, they did make a note on the cover page that said that these proposed revisions convey USP's review of and subsequent partial alignment with the ICH Q3D Step 2 limits. So the proposed limits reflect a review of published toxicology data, as well as expert review by toxicologists, and they have continued over time to accommodate ICH decision making. So despite this, there's still some divergence, but they do hope to harmonize the documents hopefully sometime very soon here. So looking at ICH, the elemental impurity limits, the first thing that becomes apparent here is the list is actually longer. So there's 24 elements in Table A21, and this is compared to the 15 elements that we're used to seeing in 232. And secondly, the elements in the ICH list are sorted by class. So this differs from 232 as well. So the 4 classes are actually grouped according to risk. So if we take a closer look at, but also some of the similarities about the table, we can see that across the top we can see the different routes of administration or dosage routes, oral, parenteral, or inhalation. Along the side we can see the elements, again quite a few more elements, and we can also see the limits there. But let's talk a little bit more about the class. So what do they mean, what does ICH mean when each element is divided up into a different class? And that actually is risk assessment based. So the ICH document does outline risk assessment strategies in some detail. So by going to the different classes, we can see that for class 1, arsenic, lead, cadmium, and mercury, sometimes referred to as the big 4, these are pretty much must be included in the risk assessment, and that's the same as USP 232. There's also the class 2A, and with these elements, you can see that these should be included in the risk assessment as well. The Class 2B, these are elements that should be included if intentionally added. The Class 3 is interesting in that these depend on the route of administration. So for example, if it's oral, it's not going to be as toxic, or these elements aren't going to be as toxic as compared to if it's injectable type drug. And then ICH also added Class 4. So this is kind of interesting because these additional elements don't need to be included in the risk assessment, and ICH states that these are commonly found at high concentrations. So there's some additional Class 4 elements that weren't included on the last table that we looked at. So I think what's helpful here is just to do a comparison side by side of USP and ICH limits. So for this example, to keep it simple, we just stuck with the oral administration route, and we're going to take a look at, let's first take a look at cadmium. So for cadmium, we can see here that the official 232, the limit's 25. In the proposed revision, it goes down to 5. And the reason we know that it went down to 5 is to harmonize with ICH. So now we have a match between ICH and 232 for cadmium. Lead wasn't as tricky because there was already a common agreement between 232 and ICH, so nothing needed to be changed. But what if we look at arsenic? Arsenic's an interesting example because here we're at 1.5 for the official 232, but arsenic is actually increasing by 10 times. So when's the last time you've seen a regulatory method where the limits increase? So that's interesting. But that was done, as we can see, to align with ICH. Mercury, on the other hand, mercury for 232 was 15 micrograms per day. ICH proposed 40, but USP said no, we're going to stick with 15. So there's not an alignment currently between ICH and USP for elements such as mercury, and we can see some other ones here as well. For molybdenum, molybdenum did change as well. Some of these other ones, like the metal catalysts, were still at an order of magnitude lower for 232 proposed as compared to the ICH. Over on the right-hand side, we can see the additional elements and the limits that are associated with those. So it might be good to note here that we've seen in some cases companies want to move forward. They're kind of tired of waiting around for the harmonization. We know that there should be some new information coming out soon, but what we have been seeing is that some customers have taken the stance that they're just going to go ahead and kind of combine the worst of both cases, if that makes sense. So for example, with mercury, they say, hey, we'll just adopt a limit of 15, be sure that we can hit the limit of 15. If it goes up in the future, that's great, but let's be sure we can hit 15. And same thing with the elements. They may also want to be sure that they can cover all these elements. That way they're, again, covered for the worst case scenario, and they can move forward with minimal risk. Okay, so now we're going to explore 233, which addresses the procedures. So for 233, the elemental purity procedures, we can see that there are 2 compendial procedures listed. Procedure 1 is Icp-Oes, and Procedure 2 is Icp-Ms. And it should be noted that alternate procedures may be used, but any alternate procedure must be validated and proven equivalent. So in this section as well, or in this chapter as well, sample prep requirements are outlined. And of course, Tim's going to be going over this in detail very shortly. And the validation requirements are also outlined in this chapter. So there's all kinds of details on specificity requirements, accuracy requirements, precision requirements, what it takes to actually validate the method. So since ICH doesn't specifically cover procedures, there was not much need in terms of harmonization revisions, but USP did take the opportunity to propose a few changes to 233, and these changes were primarily editorial and for clarification type updates. But next, let's take a closer look at the instrumentation that can be used to achieve the requirements of USP and ICH. we'll take a look at the Agilent atomic spectroscopy portfolio. And the first thing that you'll notice is that there's quite a few products in the product line. So everything from graphite furnace and flame AA, which you can see in the top left, to a unique instrument, the MP-AES, or the Microwave Plasma Atomic Emission Spectrometer. This is a product that's unique to Agilent, and it actually uses a microwave-based plasma and runs on air using a nitrogen generator. So a lot of interest around that. We can see the Icp-Oes that was released earlier this year, the 5100. Down here we see the 7900, very popular Icp-Ms, which was also released earlier this year. And again, unique to Agilent, the triple quad 8800 Icp-Ms system. So again, we do have some cases where people are exploring the alternate procedures where they might want to try and use graphite furnace for one element or try and use MP for a really specific case. But what we want to do is we want to concentrate on the compendial procedures of Icp-Oes and Icp-Ms. So first we'll talk about compendial procedure 1, which is Icp-Oes. I do just want to review the 5100 with you, and there is some new technology here that will be helpful to comply with these USP methods. I want to make sure that's understood. So first, with the new technology comes some new terminology as well. I just want to review this, is that we call this the 5100 SVDV, and that stands for the synchronous vertical dual view system. So I think everybody knows what dual view is. We can look at axial and radial at the same time, but we do want to point out that this uses a vertical torch. So inside the torch box, the torch is actually in the vertical orientation. This means that we have excellent matrix tolerance. We can handle greater than 25% dissolved solids. So that means that the sample, when it's prepared and transferred to the instrument, we can handle a really heavy sample matrix and introduce that directly to the instrument. But even with that vertical torch and excellent matrix tolerance, we can actually look at the axial view as well as the radial view at the same time. So we can maintain the detection limits at the same time as extending the linear range. We did bring forward with some changes the Vista Chip 2 detector, which has full continuous wavelength coverage, which helps us get around potential interferences in drug products. And we can also do something called qualifier wavelengths. And that's where we can look at alternate wavelengths for the same element to help make us feel comfortable and verify that our results are correct. And we have a new ICP Expert 7 software, which we'll review briefly as well. So I just wanted to pull out a couple of things that will be really beneficial to the 232, 233 methodologies. And one I already mentioned was the matrix tolerance with the vertical torch, the synchronous axial and radial viewing. Interference removal will also be important to work around those interferences that are possibly present in the product. And the dynamic range. So not only can we see relatively low levels, we can also see higher concentrations for some of these other metals that are going to be present in the drug product. So taking a closer look again at the SVDV. So here's the vertical torch, and we can see that from the vertical torch we're taking the radial view, which is the blue line, and the axial view, which is the red line. We combine those using a unique optical component called the DSC, or the dichroic spectral combiner. That's what allows us to look at the axial and the radial at the same time. And we feel that we've removed the limitations of other dual view systems in the fact that we have a vertical torch rather than horizontal, handles higher matrix, and we can look at the axial and the radial at the same time. A favorite feature of the new system as well is the simple operation torch. So you can see that you can easily insert the torch, close the torch loader, and the torch is automatically aligned. So it's a one-piece torch. There's no need for alignment. There's no need to make gas connections. It doesn't matter who installs the torch always goes to the exact same position. So this leads to excellent reproducibility time after time, regardless of who's running the system. And here we want to highlight the detector in the 5100 as well. So this is the VistaChip 2 CCD detector. On the left, you can see an expanded or blown up image of the detector. And on the right, you can see an example of what we call an oscillogram, where we can do a full frame image and look at all the different components that are present in the sample. And next, we want to talk about specificity. It is important that we're sure that we're getting the right result. One thing that we have on the system is something called fitted background correction. And this is an automatic background correction that's going to dynamically choose the best background correction points for you, regardless of the sample matrix type. So if you have varying drug products or varying components in those drug products, the instrument is automatically going to pick the best background correction points for you. And we can see an example here, low concentration of arsenic and 100 micrograms per liter of platinum that we can effectively get rid of or correct out for that interferent. The example on the right, we can see 5 micrograms per liter cadmium and that same 100 micrograms per liter of platinum that does not impact the result for cadmium. In fact, if you do have any kind of a spectral overlap, we can actually use our spectral decomposition tool where we can mathematically subtract out the spectral contribution from the interferent. So it's easy just to look at the example here for ruthenium. So ruthenium was the element of interest, and the result looked like this. It was this tall blue peak, but that result was too high. And what we found out was actually that there was contribution from chrome in there. And once we subtract out, using FACT, the chrome contribution, we can see the resultant peak for ruthenium is available. And we don't have to worry about the interferent from chrome there at 267, where typically people would have to worry about that chrome interference. We should mention we could also use ruthenium 245 if we want to, and we could use that as a backup or qualifier wavelength as well. The Agilent 7900 Icp-Ms, so this is Compendium Procedure number 2, and I think that everybody's very familiar with the Agilent 7900 Icp-Ms, a popular solution to meet the requirements of USP 232 and 233. And this has been updated, and this is building upon the previous 7500 Icp-Ms, and this was introduced earlier this year. So some of the improvements, number one, the matrix tolerance. The Icp-Ms can now handle 25% TDS. Most people ask if that's a typo. Should that be 2.5%? No, 25% TDS. So we have excellent matrix tolerance that's unheard of on Icp-Ms, and that's because of the UHMI accessory. We'll talk more about that coming up. There's also a new collision reaction cell, Octuple Reaction System 4. It removes the polyatomic interferences using helium mode. And this is nice because it's simple and easy. We can use one mode to effectively remove interferences. We'll talk more about that. There's also a new detector as well. This detector offers 11 orders of dynamic range. And in the new Mass Hunter software, there's some auto-tuning tools, predefined methods, semi-quant analysis, and HPLC, even GC coupling is now 100% Agilent. So safety, easy to use. There's also SOPs available. Okay, so taking a look at a couple of the benefits for complying with and succeeding with 232 and 233 with the 7900. First, as we mentioned, for matrix tolerance, the UHMI system. For interference removal, the Octuple Reaction System. And with the new detector, the ultimate dynamic range. So in terms of matrix tolerance, let's take a closer look at the UHMI. So UHMI is essentially a way where we can directly aspirate the sample up to 25% without dilution. So the dilution actually happens within the spray chamber area. There's an aerosol dilution. We have partial desolvation of the aerosol as well before it's introduced to the plasma. So this means less sample preparation, in terms of less sample handling, less errors associated with that, less contamination, less reagents consumption, and better detection limits. So the UHMI has been quite popular for the USP applications. Additionally, the ORS, this is now version 4, the ORS 4. The geometry is a little bit different, as we can see in the bottom left. The rods are a little bit longer, smaller ID, a little bit higher pressure, and a little bit higher frequency. So it's just really been fine-tuned to give you the superior interference removal in helium mode only. So by using helium mode only, we don't have to deal with reactive gases, so it's simple, it's effective, helium's inert, so no new interferences are going to be produced, and there's no safety issues related to reactive gases, such as hydrogen. The consumption, we're only talking about 5 mils per minute of helium, so relatively low consumption there, and we have a very fast switching of less than 2 seconds. And this means that there's no need for correction equations when using the ORS-4. To show you an example of how we can get rid of interferences effectively with Icp-Ms, this is what your mass spectrum might look like. So this is in solution. It has a couple of different acids in there. There's nitric acid, there's some hydrochloric, sulfuric, a little bit of IPA. And we should point out that hydrochloric acid will most likely be present in your samples for USP 232 or 233. It is recommended for the digestion to have an effective digestion and make sure that the sample is stable. So most likely you will have hydrochloric acid in your sample digestion. So we do need to have a way to get rid of the contribution from hydrochloric acid and these other acids. So this is what it looks like in no gas mode. You can see a really busy spectrum with a lot of interferences. But as soon as helium is turned on, all the polyatomic interferences are removed. So we can see that flattened out those interferences. So the question is, well, what about the sensitivity? Can we still see our low-level analytes in there? And we say yes. This is 10 ppb in that same matrix. So we can see there's no residual interferences. The isotope patterns match. There's no signal lost. And by doing this, having a clean spectrum, we can also have qualifier isotopes or qualifier ions. So you can look at secondary isotopes to help confirm your results. The reason we can do that is because you now have a clean spectrum. And here we can see the 7900 Icp-Ms calibrations for the big four. So as mentioned, the big four are arsenic, cadmium, lead, and mercury. So the 7900 has no problems calibrating at those very low levels, but with a new detector, it's a new proprietary and patented detector, it's called the ODS, the orthogonal detector system, we can actually see 11 orders of magnitude dynamic range. So we can see these ultra low levels that you see here, but at the same time, we can pick up the higher concentration elements, all in the same run. Additionally, there's a semi-quant analysis mode for helium. So here we can pick up all the elements of interest. And again, because of helium mode, we're removing all the polyatomic interferences, and we can screen complex matrices with ease. So I just wanted to kind of summarize a couple of those things, the Agilent Icp-Oes and the Icp-Ms, the unique capabilities that help us comply with 232, 233. First of all, run samples with minimal handling following sample prep, and this includes high matrix samples. So for the Icp-Oes, we do that with a vertical torch. On the Icp-Ms, we have the UHMI. Common methods can be used across multiple matrix types or across multiple drug types. And this can be done on the Icp because of fitted background correction or FACT spectral deconvolution. On the Icp-Ms, the octuple reaction system with helium eliminates the polyatomic interferences regardless of matrix type. And results confirmation. This is something that's becoming very popular if customers can have additional data that supports the results. Obviously, they're going to be all for that. So on the Icp-Oes with full wavelength coverage, we can do that. And on the 7900 we have access to those interference-free qualifier wavelengths as well. A couple other things, the 5100 we can collect, and the 7900 we can collect all the 232 and ICH elements during the sample analysis. So in some cases, people are deciding that they are going to use the risk-based approach. They're only going to look at certain elements that they need to look at. Other customers are saying, hey, if we have one of these two tools, we can actually look at the complete list, the complete USP list, the complete ICH list, and look at everything at the same time. And validation, compliance, and training solutions are available on both the Icp-Oes as well as the Icp-Ms system. So comparing a little bit the Icp-Oes and Icp-Ms, how do you choose or how do you pick? We should say that detection limit suitability is going to be a key concern. So exposure factor and daily dose, depending on the exposure factor, depending on the daily dose, those can drive the detection limits lower. And also the dilution from digestion. If you do have to do a larger dilution during the digestion process, then you're going to end up with lower required detection limits. Speciation capability. If speciation capability is desired, Icp-Ms is probably going to be the tool of choice because we can easily do speciation on the 7900 Icp-Ms. And really, you just need to consider the overall organizational needs. Is this an excipient company? Is it an API or drug product manufacturer? Is it a static production facility that looks at one or two products, or is it more of a dynamic QA and R&D lab that has many products? But what we are seeing is that a lot of people are considering future needs and the potential for changing limits. So we can kind of think of this as insurance for the future. If you want to have the lowest detection limit capabilities moving forward and ensure compliance in the future, people are looking for Icp-Ms in that case. If you're comfortable with the limits that are currently proposed or you feel will be coming out, then the Icp-Oes may be appropriate. Of course, budget's going to come into play there as well. So I want to run through quickly an example of calculating the J value. So we're going to look at 3 different examples here. And this is an interactive calculator that we have available. There's going to be some assumptions here. The first assumption is this is an oral daily dose. So the limits will be slightly higher because of that. And we have to put in our dilution factor as a result of the digestion procedure. So for this example, the digestion procedure was 1 gram into 50 mils. So we can see that yields a 50x factor. And so that's going to drop our level down to 0.1 micrograms per day once it's adjusted for the prep. And then we also have to insert the maximum daily dose. So if the maximum daily dose was only 1 gram, we can see that the J level for cadmium is going to be 0.1, for lead 0.1, arsenic 0.3, and mercury 0.3. But those that are familiar with the method will know that we have to be able to see accurately at 0.5 J. So we then need to cut that level in half so we can see the required detection limits for this particular scenario. So next, what happens if we make a change? So while currently we're using the factors of 50 and 1, what if we do a more concentrated sample prep and the dilution factor is only 25 times after the digestion procedure? We can see the effect on the detection limits. The detection limits, or the J level, the required J level, actually increases a little bit. But for the third example, what if we increase the maximum daily dose? So here we're sticking with the 25 times dilution factor from the digestion, but we're increasing the daily dose to 5 grams. You can see that's going to significantly lower that 0.5 J level. And we should keep in mind, if you are going to go above 5 grams, what if you're going to be pushing into 10 grams or higher? That's really going to lower the 0.5 J level even further, and that's where you're going to start to be outside the realm of Icp-Oes, and Icp-Ms would be the obvious solution. Again, if speciation is required, you do want to look at Icp-Ms. So 232 does reference inorganic arsenic. So inorganic arsenic, if you do exceed arsenic, it could be speciated, and if you prove that the inorganic component is less than the limit, then the drug product is still going to be in compliance. So we can see the 7900 with the LC system here. And then we do have a number of publications, so elemental impurity analysis publications. These are all available from the Agilent website. Application notes, regulatory compliance notes. There's a primer on USP. Agilent also has industry-leading compliance solutions. So we have compliance solutions that are going to meet the needs of the regulatory authorities, the FDA, 21 CFR, and these include system design qualification, quality systems, the software validation, and these are available for the Agilent products. The compliance software support information, we know that this is really an important part of integrating a new technique to the pharmaceutical laboratory. So we've really gone into great detail to make sure that all these things are in place and make sure from the beginning of the analysis through the reporting of the data that you're well covered. And next, let's look at that proposed timeline. So some of these dates are already passed. So February 2013, the 232 and 233 became official, but not implemented. And then in July 2013, ICH releases the Q3D Step 2B document. In December 2013, as we know, revisions to 232 and 233 were proposed with changes to partially align with ICH Q3D Step 2B. And the big ones that are coming up, September 2014, it is expected that the Q3D guideline will reach Step 4 by the end of September of 2014. And this is some meeting report notes from the most recent ICH meeting, which took place in June in Minneapolis. I know many of you are saying, wait a minute, this is September 25th. So hopefully we'll see that document very soon. And then USP has stated that on August 1st, 2015, they expect to have the revised 232 and 233 become official. And then on December 1st, 2015, revised 232 and 233 are going to be implemented. And this will include removal of all references to 231 from the monographs. So that date is next year, but it is coming up very quickly. I think people are starting to realize that what they're going to have to do is they have to purchase the instrument. In some cases, they have to train the analyst. They have to validate the products. So when they start thinking backwards in terms of how much time this is going to take, they realize that December 1st, 2015 is coming up very quickly. So please let us know what we can do to help you with any of the decisions that need to be made around implementing these methods in your laboratory. And at this point, I'd like to thank you and turn it over to Tim. He'll talk a bit more about 233 and talk about important considerations related to microwave sample prep technology. Tim?
Great. Thank you, Steve. That was a great summation and review of the methods as well as the technologies that Agilent has. So in this portion of the presentation, we're going to switch gears, obviously, and talk about the sample preparation component of USP 233. And although there's only a couple paragraphs really in both methods regarding sample prep is a key consideration and a very key component in making sure that the data quality and the data that you achieve or get from your ICP or Icp-Ms is a top quality. So we're going to talk about 233. We're going to review microwave digestion and some of the key considerations as well when approaching microwave sample prep. We're going to talk about a relatively new technology in the area of microwave digestion called single reaction chamber technology, or SRC, and then do a real brief review on the different microwave technologies that are available from everyone out there. Okay, so USP 233. Now, obviously, Steve spent a lot of time on the measurement component of that method. And from the sample prep side, we actually are given several choices of the sample prep technique that we're going to use. And these choices include neat. And if we're lucky enough to run the samples neat, basically all you have to do is inject the system right into the ICP or the Icp-Ms. You also, if your samples are suitable for this, if they can be directly dissolved in either an aqueous or an organic solvent. That is also applicable and open to be used. And then you also have the indirect solution technique for sample prep. And that is required when your material or your sample types are not directly soluble in a solvent system of some kind. And here we would be doing total metal extraction using a closed vessel digestion procedure. Now, I'll know that USP does not specify closed vessel microwave digestion as the technique to use. A lot of labs are going this route, and the reason for that is, microwave digestion is a very mature technology. It's been around for a long time, and for good reason. One is, the digestion speed and the digestion quality are very high. In a microwave, the key concept is that you're working inside of a sealed vessel. By working in a sealed vessel, you work at elevated temperatures which both increase digestion time, or rate, let's say, as well as digestion quality. And for USP, that's important. Drug products are composed of a variety of different matrices. Microwave really provides maximum flexibility to you. In microwave digestion, we also minimize the digestion blank, definitely a critical component of trace metals analysis in general. Small amounts of acid are typically used, anywhere from 5 to 10 mils to digest your samples, and you have much better control of outside contamination, again, because you're working in a sealed vessel. Finally, microwave often gives you the best retention of volatile elements. Two of the class 1 big 4 elements, arsenic and mercury, for example, are very volatile, and microwave is a very solid approach for getting good data for these elements. Now, when you're looking at microwave, right, there are a lot of different options available to you, and I'm sure that as many of you have gotten into this, there's a lot of different information out there, and it's how do you figure out what is really important to determine what is it that I need to do? What is the technology and the configuration that's right for me? Well, if we take the overall idea that successful sample preparation really depends on selecting, 1, the correct acid chemistry, 2, figuring out the correct temperature that is going to give you both proper extraction of your elements of interest, and that's going to create a homogeneous solution. Now, what we first must do is understand our sample type and understand the temperature that we need to obtain to get a good digestion, as well as the associated pressure that is going to be generated when we're digesting that sample. Or once this is understood, you can then select the different microwave vessel that might be appropriate for you, or the different microwave configuration that is best based for your needs. The reason that this is important Okay, everyone. Sorry about that excitement there. Technology at its finest. We seem to have been dropped from the presentation, but I believe that everyone can hear me now. So to jump right back in where we stopped discussing microwave sample preparation and choosing the right vessel, really it's imperative to choose the right vessel that can handle the temperature and pressure needed to successfully digest your sample so that, one, you can get complete digestion of your sample, and two, you can get full retention of your volatile elements. If you selected a vessel that was not capable of achieving those temperatures and holding the necessary pressures, you could have problems getting good data from your samples. Now, to give you just some visuals about what I mean by this, this was ginkgo biloba, and we digested this sample at two different temperatures, 180 degrees C as well as 200 degrees C. Okay? Now, we took a half a gram sample for both digestion temperatures, decanted after the run, and diluted up to volume. This is the sample here that was digested at 180 degrees C. During this digestion, we achieved a max pressure of 32 bar. What I want you to see on this picture is that you have this very yellow type color, right? For us, what we're looking for after dilution, we want a clear solution. This tells me that we haven't broken down the complete organic backbone of this sample. To do that, we can use the same acid chemistry, but we need to bump up the temperature. When that sample was digested at 200 degrees C and diluted the volume, notice the nice, clear solution that was obtained, and also know the pressure that was generated, 45 bar. Now, yes, you're going to have some increase of pressure simply from the vapor pressure of the acids that were being used. In this case, it was nitric acid. But that additional pressure is coming from the further decomposition of the organic component of that sample. To look at another sample, an even more difficult one, high-density polyethylene. This sample we digested at 180 degrees C and 220 degrees C. Same sample size, half a gram for both digestion temperatures. At the one digested at 180, you can still see sample inside, okay? It is not a complete digestion, nowhere close. And you can also see that the max pressure was only 20 bar, okay? So there was a lot of organic component that still need to be oxidized and broken down to get a good, clear solution like the one that we see here at 220 degrees C. Notice that when we digested a half a gram of high-density polyethylene at 220 C, the pressure generated was 58 bar. That's pretty substantial. So if we had to digest something like high-density polyethylene, and there's a lot of different sample types for USP that are somewhat similar in nature, being that they're both organic, so they're going to generate pressure, but they're relatively stable compounds. If we had a vessel technology that could not hold 58 bar of pressure, we would never be able to successfully digest that sample. It's just not possible. So going back to my original statement, one of the key considerations to make is that you first understand your sample type, the temperature needed to digest the sample, and the pressure that's going to be generated when that sample is digested. And certainly your microwave vendor is capable of helping you all figure that out. Now for USP 233, there's some general recommended procedures, certainly that can help get everybody on the right track. Sample size is typically about 1/4 gram, but certainly that can be modified due to your specific detection needs, perhaps homogeneity issues. You might need a larger sample to get a more representative sample, as well as the microwave vessel technology that you have. From a standpoint of acid chemistry, nitric acid, or most commonly or most often a combination of nitric acid with a little bit of HCl, is going to be the typical acid mix that is going to be used. HCl, why? Not necessarily because it's going to help with the actual digestion of the sample, but because it's there to help stabilize certain elements. Mercury, platinum group elements, etc., really benefit from having some chlorine in the solution. In regards to HF, unfortunately, it doesn't matter how high a temperature we go inside of a microwave. Chemistry is still chemistry. So if you need a complete digestion and you have silica as well as titanium or possibly tungsten in your sample, HF is going to be needed to get a nice clear solution. Unfortunately, there's just no way around it. With that being said, we have had some luck with some alternative chemistries to HF, one of them being tetrafluoroboric acid, or HBF4. The couple nice things about using this acid is, one, it's not nearly as, it's still a concentrated and aggressive acid, but it doesn't have the safety concerns that HF has surrounding it. The other nice thing about fluoroboric acid is that it helps with insoluble fluorides. Sometimes when you're using HF, or often when you're using HF, and you have elements such as aluminum, magnesium, and calcium at fairly high levels, insoluble fluorides are going to form. And what you typically have to do if that happens is that to get a clear digestion, you're going to have a second step in that process where boric acid would be added. Okay, and that's to help neutralize the HF. Here with fluoroboric, it's one step, and again, you don't have the safety concerns that you do for when you use just HF. Another consideration here needs to be made is for osmium. Osmium is one of the elements that possibly needs to be analyzed for if it was intentionally added to the sample, most commonly as a catalyst. Osmium is challenging because in an oxidizing environment, it becomes extremely volatile. So to prepare that sample and analyze for osmium, most commonly as of today, the most widely used application for it is by digesting that sample in dilute HCl. So, you know, if you have an organic sample where you have to analyze for osmium, currently what is going to have to be done is that you're actually going to have to digest that sample twice. One in dilute HCl for the osmium analysis, the other using nitric acid and a little HCl, as I mentioned before, to break down the organic component and analyze for the big four and all the other elements that may be of interest in that sample. With that being said, certainly this is, given the challenge of it, and a lot of labs in the industry are interested in it, further testing by both Milestone as well as Agilent will be performed here, and we hope to find some additional methods for doing, more easy methods for doing this method. Finally, for doing sample prep for USP 233, digestion temperature. Temperature is really matrix specific, but most digestions are going to be conducted somewhere between 180 or 200 degrees C up to 250 degrees C. Really, again, depending on your sample type. And again, microwave vendor can certainly get you a long way there in selecting your proper microwave method to be used. So, coming back to closed vessel microwave digestion, again, it's going to be the primary closed vessel technique for USP 233 for all the reasons that I mentioned before. However, with traditional benchtop microwave digestion systems, and here you see our Ethos-EZ, and traditional benchtop microwave systems I describe as using individual pressurized reactors inside of a large cavity, and these are available from all microwave vendors out there. There are some inherent limitations in these. One, you're doing your digestions in Teflon vessels, most notably, which have temperature and pressure limitations. There's also a fair amount of sample handling or vessel handling, let's say, that goes into using these vessels, capping and uncapping, and there's also 5, 6, 7 components to assembling and disassembling these vessels. Another limitation of traditional benchtop systems is that you have to batch like samples together. There's no way of actually controlling the temperature in every vessel in these systems. All those samples are exposed to the exact same microwave energy, and they're all going to react differently. Different acid chemistries absorb microwave energy differently. Different samples go exothermic at different temperatures, etc. So although you might be able to monitor the temperature of all vessels, there's no way of ensuring that if you're running different sample types, each one of those are going to get to your desired temperature, and no way to truly do it safely. Finally, with traditional microwave systems, depending upon your throughput of the lab and the difficulty of your samples, a lab could experience fairly high consumables costs. So with that being said, you know, in Milestone, was trying to decide where do we want to take microwave technology and what do we want to do. Well, these limitations were felt by all labs, regardless of what they're actually doing. So we thought, well, what if we could digest all the samples instead of using multiple pressurized reactors in a single large vessel that removes the temperature pressure limitations of traditional Teflon vials. If we did this, there would be a single system that for the first time would allow chemists to process any sample type, even the most difficult, in the same system. It would simplify vessel handling. It would eliminate putting the vessels together and disassembling at the time, and for the first time you could use simple, cheap, disposable glass vials. It would eliminate the need to batch samples together. It could potentially give you very fast heating and cooling because you were going to be working in a smaller chamber. And because of the labor costs, as well as the digestion times that you'd be saving, it would give you the highest throughput. Finally, this system potentially could eliminate method development almost, as one method would work for the largest variety of samples. And this is where SRC technology, or single reaction chamber technology, was born. And this can be shown in the ultrawave. Okay? Milestone introduced the ultrawave about 2 years ago. It's been received very well by the industry because it addresses those common frustrations held by anybody that's using microwave. Single reaction chamber, again, virtually eliminates vessel handling, allows you to digest mixed sample matrices, goes to very high temperatures and pressures, and really is a high throughput system. So in terms of the design of the system, it's relatively basic. There's a 1-liter stainless steel reaction vessel, and it's within this reaction vessel that you're going to be doing your sample digestions. The sample rack is lowered inside of the chamber, and there's 2 key concepts with SRC. One is that these vessels, when lowered inside of the chamber, they're actually sitting in a bath of water. We are not measuring the temperature of any single vessel, but we're actually measuring the temperature of the water. And what this does is allow us to run completely mixed sample batches, completely mixed acid chemistries, do it safely, but also ensure that for the first time those samples, no matter what's in them, will come to the desired temperature. The other key concept of SRC technology in the ultrawave is that before the start of the run, the whole chamber is pressurized, and it's pressurized with nitrogen. What this does is immediately raises the boiling point of all the reagents inside of the chamber. It eliminates crossover contamination, but what it also does is that it allows us to use very simple vials with loose fitting covers. Just a close-up picture of the actual reactor itself. Again, it's a 1-liter stainless steel reactor with a stainless steel cover. There is both a Teflon cap and a Teflon liner that fits inside, and here you have direct temperature measurement. Now, the temperature and pressure capabilities of the ultrawave are 200 bar and 300 degrees C. So when I talk about that it's applicable to virtually every sample type, it means that given the very, very high temperatures that we can achieve and the pressures that we can handle inside of the ultrawave, almost any sample matrix can be decomposed inside of the system. Just a quick plot to show you why we pressurize that system. So you all know nitric acid at ambient pressure boils at a temperature of about 100 or 110 degrees C. Now nitric acid under a pressure of 30 bar boils at 230 degrees C. And of course, during your digestion process, you're also creating additional pressure, NOx gas, CO2 gas, chlorine gas possibly, as well as just the vapor pressure of the acids at elevated temperatures. At 60 bar of pressure, nitric acid boils at 263 degrees C. So again, what this pre-pressurization allows us to do is use loose fitting covers, as well as eliminate crossover contamination from vial to vial. In terms of programming, I'll go through this slide real quickly. In microwave, you always have 2 steps, a time to temperature and a time at temperature. The ultrawave is controlled with an external touchscreen controller. All you simply add or program is your time to temperature and your time at temperature. The microwave real-time displays the current temperature inside of the reactor, as well as the pressure inside of the system, and it automatically adjusts the power so that you follow the profile created. So you don't have to change power settings depending upon the number of vessels or the volume you're running. The UltraWave automatically does that for you. Now, in terms of the sample racks and vials available, there are a couple options. The most common options that we have for USP 233 is a 15-position rack that does 15 mil vials, as well as a 5-position rack that does 5 40 mil vials. Now, these vials are available in glass, quartz, or Teflon, and again, they are simply fitted with loose-fitting caps. So vessel assembly is virtually nil. How do you choose which vial option is best for you? Well, first we start with the chemistry. If HF or fluoroboric acid is going to be needed or used during the digestion, unfortunately you're limited to Teflon. It must be used. However, if that is not the case, the second thing you're going to look at is your detection needs and your elements of interest. Interestingly enough, for certainly the big four, but most, if not all, of the elements described in USP 232, disposable glass vials work extremely well. The only exception that I would make to that is for your common environmental contaminants, sodium, calcium, aluminum, potassium, certainly silica. These elements can be found at some level, so really it's ensuring that your detection needs can withstand these background levels, and if so, glass would be available to you as well. If not, quartz would certainly also be a viable option. Okay, so almost always the first question that we get asked when we talk about loose fitting covers and doing digestions at very high temperatures is, what about crossover contamination? Do you retain your volatile elements? And certainly it's the first question I asked when the technology was provided. So to test that, what we did was very simple. We took a 15-position rack and we divided that rack in half. 8 of the vessels inside of that rack had 110 ppm mercury standard, so a traditional very volatile element. And the other half of that rack were simply blanks. We took it through a typical microwave program, 220 or 230 degrees C, and then analyzed the blanks for mercury. As you can see here, in those blanks, there was no recognizable or recorded mercury found within those samples. So the mercury is not going from one vial to the next, but what about is it staying put? Can you get 100% recoveries of the element? So to do some initial tests on this, we ran 3 certified reference materials in the system at the same time. One was a polyethylene or a polymer SRM, has a relatively high concentration of 25 parts per million. We also ran a fish protein inside of the system, relatively low concentrated sample of 400 ppb, and then also a soil. Take it through the same microwave program that we did for the blanks, about 220 degrees C, and then analyze those samples for mercury. As you can see, for each of the samples, the results were well within the limits, the ranges of the SRMs, and it, again, not only proved that we can get full retention of volatiles, but that we're not cross-contaminating. If we were, we would expect that highly contaminated or concentrated sample to impart some contamination into that fish protein. Okay, so the best way to describe the UltraWave, in my humble opinion anyways, is that the UltraWave for the first time really provides the ultimate in flexibility. There are a lot of microwave digestion systems available out there, whether it be from Milestone or others. And there are microwave systems out there specifically designed for high throughput. There are microwave systems out there designed to be easy to use. There are microwave systems designed to be high temperature, high pressure for very difficult samples. But none of these samples kind of do it all in one. And that's where the real power of the UltraWave comes from. For the first time, it's a system that kind of meets all the possible needs of a given lab. From an ease of use standpoint, here's just to give you an idea of what a traditional microwave vessel is going to look like. Okay, you have a shield and a Teflon liner, a venting mechanism of some kind, and all of these components get fitted inside of a rotor. With the UltraWave, all you have is a simple vial and a loose fitting cap that fits on top. From a size standpoint, Here's a traditional, this is actually our 10-position high pressure rotor called the SK10. From a size standpoint, again, from how easy it would be to handle, this is a 15-position ultra-wave rack. Okay. Also, as I mentioned, from a consumables cost size, as you can imagine, the ultra-wave rack is considerably less than a standard microwave rotor. So it's easy to use. That's one very big benefit, but it's also very high throughput. What we did here in this data that you see is that we took an 8-hour day, we took the ultra-wave with 2 sample racks, and we took the Ethos EZ with 2 10-position rotors. And what we did is that we digested polymer samples over the course of that 8-hour day, just one batch after another, and we recorded the time for each step. Certainly you can look at the individual times from the amount of time it takes to weigh out samples and assemble the vessels, the total run time, and why one run is running. There is time for you to set up the next run. But over the course of an 8-hour day in the ultra-wave, almost double the throughput compared to its standard system for these relatively difficult samples. Now, mind you, in the traditional closed system or in the Ethos, we are not cleaning in between runs. In a real-world environment, this would need to be done. In the ultra-wave, it wouldn't because we were just using disposable glass vials. We also did the same test digesting plants, so something like a ginkgo biloba. We did it differently because plants are easier to digest, so you can use a higher throughput style rotor. And for that, we did the same test over the 8-hour day, but using 24-position rotor in the Ethos, and again the ultra-wave we used the 15-position rack. Even so, after the end of the day, we digested 220 samples compared to 144 in the Ethos. From a workflow or an efficiency standpoint, you know, I think one challenge that many of you have in USP is that you're going to end up with possibly a very large variety of sample types. Polymers, APIs, excipients, mineral complexes of some kind. And in a regular system that uses the multiple pressurized reactors running at the same time, you're going to have to batch those together. And that's what you see pictures of here. In the ultra-wave, stick it all in the same rotor, and you're good to go. Another example of digestions that have been carried out using a very wide range of samples. This is something we did in the application lab. We did food products, pharmaceuticals, nutraceuticals, environmental samples, polymers. We did not digest the whole rubber ducky. Oils, geological samples, using different chemistries, using different vial materials, all in the same run. And again, you can digest them successfully and safely because we know that each one of those samples is going to come to the required temperature. Of course, that brings up one question that we often get. So if you're doing all those samples, how do you know what microwave program to use? Our advice is to determine which sample is going to be your most difficult, which needs the most rigorous sample preparation method. Use that one. You can't cook, or let's say overcook, the samples that digest at lower temperatures. If you use the highest one, you will ensure that you get complete digestions of the complete range of sample types. Okay, I know we're getting short on time here, so just a couple more slides. The temperature capability of the ultra-wave is extreme. There are some sample types in pharmaceuticals, especially those that have a lot of aromatic rings, that are just extremely difficult. They're extremely stable, and they really require a sledgehammer to break them down. Here's a digestion or a method that was developed for digesting triphenylphosphine. Again, has a lot of aromatic rings and is very, very stable. The only time we were able to get a complete, clear digestion of this sample is when we went to temperatures of 280 degrees C. Every time, at every temperature prior to that, what would happen is that we'd either get a milky colored solution or that it would cloud up upon dilution. When that happens, it is because the organic backbone of that sample is not broken down. The same acid chemistry can be used, but the temperature must be increased to get a good digestion. Okay, I'll skip forward here to this slide, but sample size. Pressure capability in the ultrawave of 200 bar, and sample size is important sometimes. One for detection needs, as I mentioned, or sample homogeneity, gathering a representative sample. In the ultrawave, we digested 10 grams of infant formula at one time using the 5-position rack, so 5 2-gram samples, and digested it completely without incident. In a regular digestion vessel, the max you'd be able to do is a half a gram, maybe 3/4 of a gram. So the ultrawave definitely adds extreme capability in this area. Okay, just one user profile to give you some ideas. What are our customers saying about the ultrawave? Well, probably many of you out there are familiar with Icp-Ms Health Sciences. Icp-Ms is a large contract lab with a very good reputation with offices around the world. They definitely specialize in pharmaceutical contract testing, and their sample types range from easy to difficult and everything in between, performing several thousands of samples, certainly yearly. And this particular lab was based in California. For methods that they use with the ultrawave, it's in-house developed methods, plus USP 232 and 233. And as far as instrumentation goes, they have 6 Agilent Icp-Ms and an Icp-Oes. Before the ultrawave, because of the wide range of sample types, a number of different technologies were needed. Hot block digesters, traditional closed vessel systems, something called a high pressure asher, which is for very, very difficult samples. However, when the ultrawave was brought inside, Icp-Ms was able to switch most, if not all, of their methods to the ultrawave because, again, it covered that full range of their needs. For their sample, just a little bit about the actual method. The sample amount that they use is 500 to 50 to 100 milligrams of sample, using 2 mils of nitric and a half a mil of HCl. For samples that are over 100 mgs, 4 mils of nitric and a half a mil of HCl is used. For vials, it really depends on the chemistry that they're doing and their elements of interest, so they use all of them that are available. The ultrawave is used full-time now. Just single system has increased the digestion capacity by 50 to 70%. What does Icp-Ms see in the ultrawave? What are its primary benefits? As you can imagine, it's throughput. That's a big benefit to them. But also the ability to run mixed sample batches and having a simple system to do all of their sample prep. Lower consumables costs, especially due to the fact that you can use disposable vials, and getting great retention of traditional volatile elements. Okay. So in general, certainly this is one user story, but we've developed multiple user profiles on customers that have been using the ultrawave. And really the feedback is generally the same from all of them. They love the ultrawave because it increases their throughput, has the highest digestion power. So, Steve talked about having insurance, and I think one reason why, particularly for USP 232 and 233 and labs just getting into that, the ultrawave really provides insurance because there's unknown still. It's unknown what exact samples am I going to have to analyze. It's unknown how many of those samples am I going to have. It's unknown what am I going to have to do in the future. So really with the ultrawave, it provides the most let's say, flexibility, but also the most insurance that it can cover your needs today, but also in the future. So I'm just going to finish up here real quick, last slide, on talking through the different systems that are out there today in terms of microwave digestion. And this isn't specific to vendor, it's just the types of microwaves that are available. You have sequential systems that can digest one at a time. You have traditional microwave systems using individual pressurized reactors that are both low and medium pressure rotors. I make a separate column for traditional systems that uses high pressure reactors because their capabilities are different. And then you have single reaction chamber technology. Now each of these microwave platforms definitely have a place in a given lab. Just to talk about, a few of the things. In terms of the sequential system, the great thing about it is that they're very easy to use. Snap-on caps, typically you can automate it using robotics. The challenges with it are, one, the throughput is relatively low. To digest a single sample is relatively quick, but to digest a full batch of samples can take some time. The other thing with sequential systems, or really traditional low and medium pressure systems, that it's very important that you understand your, like we talked about before, your sample types. What are the temperatures needed to digest my sample and the pressures that are going to be generated during that digestion process? It's very important that you have a vessel that can handle those application needs. Traditional high pressure systems, it's almost as we move right here, the flexibility of the systems increase. But unfortunately, the one bad thing with the traditional high pressure system is that the sample handling, the vessel handling, gets a little bit more complex, and your throughput definitely goes down. Finally, we have SRC and the ultrawave, and as we kind of talked about all through this presentation, there's a lot of benefits that this technology offers you. The one, let's say, limiting factor initially with SRC compared to others is certainly the purchase price. SRC technology is more expensive. However, over the long-term cost of ownership, labs also often get that money back because of the faster throughput, lower cost per sample, reduced amount used for both reagents, but also for consumables. So these are the different options available. Again, your microwave vendor can help you select which is best for you. So with that being said, both Steve and I wanted to thank you for joining us today. We, both companies, understand the challenges that labs are facing with the new USP procedures, again, because they are relatively new to the industry. And we both would be happy to speak with you personally, understand your sample types, your needs, your detection needs, and take you through the different systems and options available, decide which configuration might be best for your needs, but also talk about the methods so that you can get up and running as quickly as possible. So with that being said, I'm going to take a couple minutes, and we ran a bit longer than expected, but I'll take a few questions here and then pass it over to Steve. One question, any contamination from the caps? And I assume those are the caps on the individual ultrawave vials, but maybe also as well as the reactor. So interestingly enough, the real reason we have caps on the individual ultrawave vessels has nothing to do with actually keeping material inside the vial. It's actually keeping water from entering the vial and diluting the sample. So what happens with microwave, because of how it works, you actually have reflux that occurs inside of the digestion chamber. And that reflux those vapors will condense on the cap of the reactor and potentially drip inside of the individual vials and diluting the sample, possibly giving us incomplete digestions. From a contamination standpoint, no, we have not seen that, although I would recommend for the individual vials, cleaning them in between digestion runs. Cleaning them would be just a simple soak. Many labs just have a beaker of very dilute acid sitting inside of the fume hood that they let the cap soak in between runs. In terms of the cap on the actual inside of the chamber itself, the large one, no, there is no contamination that would be coming from that area. How would you recommend cleaning reusable Teflon vials between runs? Great question. The one challenge with using Teflon, and for all of you who have been using microwave from some time already know, is that Teflon is great for doing sample preparation in. It has low impurities, it's resistant to acid attack, etc. The challenge with Teflon is that at high temperature and pressure, you can get contamination stuck inside of the pores of the system, and when it cools down, they get trapped. So really the best way to clean your reactors, the most thorough way of cleaning the reactors, would be to actually run them as blanks inside of the system. Now, with that also being said, my recommendation is to try several different means. I do know labs, particularly for doing USP type elements and detection needs, that do also a basic acid soak for these vials, and they work as well. The last thing that would be available would be what we call an acid, basically an acid dishwasher, but it's a system actually Milestone has called the Trace Clean. You have Teflon rods inside of a closed system, hot acid vapors created that condenses on the vessels, and basically it's an acid wash, but there's really no acid soak, so no cleanup and no, let's say, hassles that are typically associated with that. Great question. Is total digestion a must? No, it's not. The only way, of course, that we can be completely sure that we have everything into solution is by getting a complete digestion. However, USP does allow for using alternative methods as long as they've been validated and shown to work. So some labs, you know, again, I talked about silica and so forth earlier. Some labs would prefer not to use HF, so they very well may be just using a nitric and HCl bath, or sorry, digestion chemistry to digest their samples and get excellent recoveries of their elements of interest. Okay, I'm going to pass this over to Steve now. There are a lot of questions. Again, we will be sending everybody a copy of the presentations as well as a list of the Q&A. For those of you who didn't respond to, Steve and I will also be responding personally to you. So your question will absolutely be addressed. Thanks once again, everybody. Here's Steve.
Thanks, Tim. I'll just go over a couple of questions here real quick that came in during the presentation. And some of these had already been addressed, but I guess they just wanted some clarification. So number one question was, when is ICH Step 4 announced? And again, the latest we have on that is from the meeting report notes from the most recent ICH meeting, which took place in June in Minneapolis. And the notes that came out from that said it's expected, this is a quote, it's expected that Q3D guidelines will reach Step 4 by the end of September 2014. So expected to happen anytime, so stay tuned for that. We're all anxiously awaiting that. Another question was on compliance solutions for instrumentation. Specifically, is the 7900 Icp-Ms compatible with my OpenLab ECM system? So yes, OpenLab ECM can be used with the 7900. There's a couple other compliance solutions as well for those that don't have OpenLab ECM, but the answer is yes, it is compatible. Number 4, the other one is, I think this question came in early on, so hopefully this got addressed. Which value do I need to hit when testing? So of course that's going to depend on a number of things. If we just make some assumptions, if it's you know, we need to, if it's a drug product, of course, and if you're doing the drug product analysis, we need to consider things like the route of administration. Is it oral? Is it injectable? We have to apply that factor. What is the daily dose, if known? That should be factored in there as well. And something that's not really clearly mentioned in examples in USP or ICH is be sure and take into account that prep factor. So if you have a solid product and you digest it, so let's say that you put 1 gram into 50 mils, you do need to account for that 50 mil dilution. I think most people understand that, but it's not really clear with the examples that are given in some of the documents that are out there. And then along those lines, there were a couple of requests for the J value calculator. Is that available at the Agilent site? It's not actually on the Agilent site, but please just feel free to send an email. My contact information is on the screen right there, and I can email you that interactive J value calculator. With that, I think that is going to kind of wrap it up for now. If there's any questions that didn't get answered, as Tim mentioned, we'll follow up with answers to those questions shortly. And we just want to say again, thank you very much for sticking with us and talking about the upcoming USP regulations. And please be sure to let us know how we can help with you implementing these methods. Thank you.